So Chapter 11 is on cell communication, and this is part 1 of the lecture. So cell signaling is what we mean when we say cell communication, and its purpose is to coordinate activities between the cells. So for example, with the simple organism of yeast, they use cell signaling for mating purposes. And all cell signaling comes down to what we call a signal transduction pathway. A signal on a cell's surface needs to be converted into a response, and that usually is going to occur through a series of steps. Earl Sutherland broke down the stages of cell signaling into these 3 steps: reception, transduction, and response. So no matter what the method of signaling that is occurring, we can really break down the type of communication into these 3 specific responses or steps in terms of getting to that response. You can see in the diagram down below here, reception is usually going to occur on the cell membrane. Transduction will occur within the cell itself, and the response also will occur someplace within the cell. There are basically 2 types of signaling that occurs, or different types of communication, either local or long distance. So in both types of communication, the target cells need to recognize and then respond to a specific signaling molecule. So that's why in a local communication, we have what we call a local regulator. And that's defined as a substance that influences cells in the same vicinity or same area. And so examples of this are growth factors or neurotransmitters. We call a local type of signaling a paracrine signal because these are chemical signals released into the extracellular fluid, the fluid surrounding the cell, and then adjacent cells that are close by respond to the local regulator. And the cells that are being targeted are called the target cells. A lot of times paracrine signaling is only going to happen if a number of cells simultaneously receive and then are able to respond to the signal. So an example of this is again with growth factors. Growth factors will be released into an area, and then a number of cells will respond to those growth factors. Synaptic signaling is another type of local communication, and that is specifically referring to neurons. So a nerve cell releases neurotransmitter molecules into the synapse space, which is that narrow space between the transmitting nerve cell and the receiving target cell. And that space is almost allowing those 2 cells to touch, but there's still a gap there. And so there is a signal that travels over that gap, that synapse, and that signal molecule is a neurotransmitter. The only type of long distance communication that occurs is going to occur with the help of hormones. So it's called hormonal signaling, and this results from specialized cells that secrete the signal molecules, which are hormones. The secreting cells are called endocrine cells, and they release these chemical signals into the bloodstream. And the bloodstream will then take the hormones throughout the body, and the goal is to coordinate different parts of the organism by reaching their target cells and getting the response that's needed. Another type of communication that it seems like it's a local type of communication is really direct contact. So cells can communicate when they are in direct contact, and they do that through cell junctions, so that substances in their cytosol can pass freely between the cells. So it's almost as if there is an open door between the 2 cells It's because the cells are actually touching each other. They can also communicate with the contact of molecules on their surfaces. So I want to talk about some of the different types of direct contact that can occur, some of those junctions. So the most common one is called a gap junction, and that's shown here in this picture. A gap junction is when there are actually channels that connect two cells. And those are they're very tiny channels between 1.5 and 2 nanometers. And these allow that free passage between the cells of ions or small molecules, nutrients. And the junction is actually a cylinder made out of proteins called connexins. Kind of makes sense. They connect. Another type of a kind of a similar idea in a plant cell is called a plasmodesmata. And a plasmodesmata is basically the same idea where these two cells are actually joined together. They're in direct contact, and there are again open spaces between the two cells and their membranes that allow the flow of molecules between them, the two cytoplasms or cytosols. Now, another type of junction that can occur is called a tight junction. And that really is not so much a communication type of junction. It's really has more of a functional reason for existing. So a tight junction is when adjacent cells are so close together, they actually end up limiting the passage of molecules or ions between them. And so this serves a function in particular with skin cells, so epithelial cells, both on our skin and within our lungs, for example. It really limits the passage of the molecules and ions from passing into our bodies, for example. So with epithelial cells, their job is to protect the body. They're the first line of defense in the immune system. And so these tight junctions actually force anything that's trying to enter the body to have to do it through diffusion or active transport into the epithelial cells themselves. So things are forced through the plasma membrane of epithelial cells rather than trying to squeeze through between the cells. So that's the purpose of a tight junction. In a similar way, there's something called an adherent junction, which is found in cardiac muscle, and that is actually mechanical attachments that actually hold cells together. And for example, in cardiac muscle, that keeps the cells together as the heart's expanding and contracting. So it's almost like sewing them together. They also hold epithelial cells together. Where this gets into communication is these types of junctions, adherent junctions, actually seem to be involved in what's called contact inhibition. So cells have the ability to communicate with each other through contact. And when there is contact that actually sends a message to the cell to stop dividing or to stop growing, that's called contact inhibition. So, for example, as cells are going through mitosis and multiplying, when there are enough of them together, there's contact inhibition that tells them stop dividing, stop making new cells. So this is an important thing. And the reason we see that it can go wrong is in the example of cancer. So in tumors, what happens is something is interfering with that contact inhibition so that cancer cells don't get the message or tumor cells don't get the message to stop growing through that contact inhibition, which is why a tumor set of cells will start like growing and multiplying on top of each other So that's how you get that sense of a mass. And you can see that especially on tumors that are found on the skin. They do begin to mass up instead of making this nice, even plane of cells that have stopped multiplying when they fill out the space they're trying to cover. Okay, so back to our signal transduction pathway. The first step of that was reception of the signal, which begins the transduction process. So reception is when a chemical signal binds to a cellular protein, usually found on the cell's surface, but it can also be found in what we call an intracellular space. So we have intracellular receptors actually found within the cell's cytosol or within the nucleus of a cell. But most receptors are going to be found on the plasma membrane. And similar to the idea again of enzymes and substrate, a signal molecule is going to fit a specific receptor's shape and attach there. That signal molecule is called a ligand. A ligand is that small molecule that is specifically bound to a specific receptor. Let's see. I think that's it. Oh, well, the other thing I wanted to add with that is the intracellular receptors, the ones that are found within the cell, primarily those are going to be, in terms of being the first point of signal reception, the ligand that's going to attach to those is most often a hormone. And the reason is that steroid hormones are small lipid soluble molecules. And because of that, they can pass through the phospholipid bilayer of the membrane. So they don't need to stop at a receptor, a protein receptor on the outside of the cell. They actually can pass very easily through the cell membrane and find a receptor inside the cell itself. So it makes a much quicker response time. So we're going to go through the three major types of membrane receptors. The first one is a G protein linked receptor. And so this works with, of course, a G protein. It's the largest family of cell surface receptors, and they're found in all eukaryotic cells. For example, they're responsible for fetal development. They're also very much involved in our vision and smell, but specifically in humans, and they depend on these proteins. There are about 1,000 different G protein receptors and ligands that are responsible for the sense of smell. So first of all, the structure of this G protein receptor is very specific. So I just mentioned there are 1,000 different ones in terms of the function of smelling, but each of them shares the same structure. So a G protein receptor consists of one long amino acid chain, or so a polypeptide, that actually winds back and forth across the plasma membrane to make exactly 7 alpha helixes that are transmembrane channels. So the way this works is a G protein receptor will get a ligand that binds to that receptor. And when it does, the receptor proteins go through a conformational or actual physical change that then allows them to activate the G protein that's attached on the cytoplasmic side of this receptor. So here's our receptor here, which is that 7 trans channel proteins. It's actually one protein going through the channel 7 times. but attached to it is this G protein. So